I'd like to thank the organizers and my colleagues at Tsinghua University to invite me and give me this opportunity to share our work with you. So my lab works on, traditionally works on bacterial-host interaction. In today's topic, I mainly talk about a cell death called pyroptosis, which is mediated by a family of protein called gasdermins. So gasdermins are pore-forming proteins. When they become activated, they will drive cells into pyroptosis. And this cell death is a pro-inflammatory cell death, plays an important role in antimicrobial defense and sepsis. So I'm more on the host side to study bacterial infection. So I first would like to show you what is pyroptosis, the morphological changes. As you can see from this movie, pyroptotic cells, they will swell, eventually undergo burst with membrane lysis. And that will release cytosolic contents and trigger strong pro-inflammatory response. And this morphology is quite different, distinct from the traditional apoptotic cell death. As you see, apoptotic cells, they will shrink and maintain plasma membrane integrity and form this apoptotic bodies with PS flipping from the inner leaflet to outer leaflet that serve as the eat me signal for macrophage to engulf apoptotic cells. However, for pyroptosis, we have swelling and membrane lysis and release cytosolic contents. So you can see that morphologically and function-wise, apoptosis and pyroptosis are on the opposite. And pyroptosis was originally discovered in bacteria-infected cells. Back to 1986, Arthur Friedlander in the US and noted that if you treat mouse macrophages with anthrax lethal toxin, and the cells will undergo a lytic death. At that time, it was not called pyroptosis, but now we know it's pyroptosis. Many invasive bacterial pathogens like Salmonella typhimurium, Shigella flexneri will also induce macrophage to undergo pyroptosis. It's a host defense response. The cell death is a host defense response, stimulate pro-inflammation for antibacterial defense. In 1996, Arturo Czerklinski, working on Shigella flexneri, discovered that caspase-1 is absolutely required for pyroptotic cell death induced by Shigella infection. So what is caspase-1? So caspase-1 is downstream of this so-called inflammasome pathway. So inflammasome are key innate immune mechanisms. They are cytoplasmic multi-subunit complex. And within the complex, we have null-like receptors to recognize bacterial infection as well as other immunological stimuli. So in 2011, we discovered this NAP-NRC4 inflammasome that will recognize bacterial flagellin. And pyrin is another inflammasome will recognize bacterial toxins that modify Rho GTPase. And the best known canonical inflammasome is the NRP3 inflammasome, which is connected to many cellular processes, including bacterial infection. So inflammasome will sense bacterial infection, drive caspase-1 activation. And back to a few years ago, my lab and Vishwa Dixit's lab at Genentech, we discovered another pathway, so-called non-canonical inflammasome, mediated by caspase-11 in mice and caspase-4 and 5 in human. And those caspases do not require null-like receptors. Instead, they directly recognize cytosolic LPS and it gets activated. So in 2015, we discovered another protein downstream of caspase-111 called gasdermin D. So gasdermin D is a 500 amino acid residue protein with 2 domains staying in an auto-inhibited state. The inflammasome activated caspase-1 or caspase-11 will cleave gasdermin D in the middle linker region, and that will liberate the N-terminal domain of gasdermin D. And this N-terminal domain, when encountering membrane lipids, binding to membrane lipids will undergo dramatic conformational change and oligomerize into a pores, get inserted into the membrane. So here's the enlarged view of this gasdermin D pore formed on the plasma membrane upon cleavage and activation by caspase-1 or caspase-11 downstream of the inflammasome pathway. Here is a beta barrel that gets inserted into the plasma membrane. So for each pore, you have 27, 28 protomers, which is the N-terminal domain of gasdermin D. So the pore is quite large, as you can see, it's 22 nanometer. And actually, before the cells undergo burst, the pore can serve as a conduit for I-1 beta and IL-18 to be released from the inflammatory cells, because caspase-1 was first discovered as an enzyme to process interleukin-1 beta. So for the non-canonical pathway, it seems that the caspase only cleave gasdermin D back to a few years ago at that time. So as you can see, this non-canonical pathway, caspase 4,5,11, recognize LPS leaking from the intracellular bacteria. So this pathway plays a key role for antibacterial defense. And within this pathway, there is another family of protein called GTP binding, interferon gamma induced GTP binding proteins. And those GBPs, they will surround the intracellular bacteria and attack the cell wall and cause cell wall stress to facilitate the release of the PAMs, LPS, or DNAs from the bacteria. And then those PAMs, including LPS, get recognized by caspase 4, 5, or 11. And so this pathway, for a successful human pathogen, the pathogen will try to deal with the pathway, try to shut down the pathway, or avoid being recognized by the pathway. So back to quite a few years ago, we showed that Shigella fraxinare has a ubiquitin ligase type 3 effector, IPH 9.8, which will catalyze the ubiquitination of GBPs and ship the GBP to proteasome for degradation. And a few years ago, we discovered another bacterial type 3 effector called ASP-C3, which will post-translation modify caspase 4,5,11 and completely inactivate the caspase to block the pyroptotic pathway. And work from Vishwa Dixit's lab show, and the pathogen has another ubiquitin ligase, IPH 7.8, which will ubiquitinate gasdermin D directly to, and degrade the pore-forming protein to prevent activation of pyroptosis. So, and this suggested that, to highlight the importance of this non-canonical inflammasome-mediated pyroptosis for antibacterial defense. So in the next about 20 minutes, I'll show you summarize two stories to you about this non-canonical cytosolic LPS-induced pyroptotic pathway, and it operates in epicellular cells and brain endothelial cells. And the reason I want to talk about these two stories is because traditionally the inflammasome pathway, caspase 1, NRP3, NRC4, they mainly operate in myeloid cells. They mainly operates in myeloid cells. As you can see from this panel of epicellular cell line profile, we profile the expression of the inflammasome components. You found that caspase 1 and I1 beta, they are rarely expressed in those non-myeloid cells, consistent with the known established knowledge in the field. However, if you look at the caspase 4, gasdermin D, this non-canonical pathway for detecting cytosolic LPS, you will find that they are widely expressed across the different epicellular cell lines. And in addition, we also found that the pro-IL-18 is also widely expressed in those cells. And traditionally, IL-18 is a cytokine downstream of caspase 1. However, in those cells, you can see that they express pro-IL-18, but they don't express caspase 1. So that doesn't, so the established knowledge or the dogma in the field does not explain how IL-18 gets processed in those cells, because they don't express caspase 1. So we came out with a hypothesis that maybe IL-18 is downstream of the non-canonical inflammasome pathway upon activation, upon detection of cytosolic LPS. So we took those cells that are positive for caspase 4 gasdermin D expression, and we just transfect the LPS into the cytosol. Of course, we get a pyroptosis because caspase 4 will recognize the cytosolic LPS and cleave gasdermin D to drive cells into pyroptosis. And we also see IL-18 secretion by ELISA assay, where you can see that pro-IL-18 gets converted into the mature form. So this data suggests that those cells do sense LPS by caspase 4, and then IL-18 become processed. If you do Salmonella typhimurium infection, you basically get the same results. And in vitro, if we took active form of caspase 4 to cleave a pro-IL-18, you can see very efficient conversion of pro-IL-18 into the mature form. And the cleavage efficiency by caspase-4 or LPS-activated caspase-4 is slightly higher than the canonical enzyme caspase-1. However, for the mouse protein caspase-11, which is the autolog of human caspase-4, both caspase-4-11 can recognize cytosolic LPS cleave gastromin D to induce pyroptosis. However, mouse caspase-11 barely cleave mouse pro-IL-18. So that's why this pathway has been missed for a long time, because the immunologists usually only work on a pathway that is conserved in the mice. So this data suggests that the dominant pathway for IL-18 activation is not the caspase-1 pathway operating in myeloid cells, but it's the LPS-activated caspase-4 and 5, mainly in the epicellular cells. So we basically put IL-18 as a cytokine target that will link caspase-4-5 mediated pyroptosis to type 1 immunity, because IL-18 has a strong activity of inducing interferon gamma production and stimulate type 1 immune response. In collaboration with Dr. Jingjing Ding's lab at Chinese Academy of Science, we co-crystallized the caspase-4, active form of caspase-4, in complex with pro-IL-18. So all the active caspases are dimer, as you can see. And once the dimer is formed, you see this little beta sheet in the active caspase-4. And this little beta sheet will recruit bind pro-IL-18 and then put the cleavage site into the catalytic pocket. So this enzyme substrate recognition mechanism is reminiscent to caspase-4 recognition of gastromin D, so the other substrate of the cytosolic LPS receptor, caspase-4. So they also, the active caspase, inflammasome activated caspase-4 also use this little beta sheet to recruit gastromin D. And the structure also explains why caspase-11, the mouse protein, doesn't work. So in the structure, in the caspase-11 structure, there is a little beta conformation, a deviation of this L4 loop that will destroy or prevent the little beta sheet to bind the pro-IL-18. So we are now taking advantage of the structural knowledge to generate mutations in caspase-11 that will gain the activity of processing pro-IL-18. So we are now making humanized mice so that we can revisit the immunological function of IL-18 using the mouse system, including the role of IL-18 in antibacterial defense. So this pathway clearly is important for antibacterial defense. And we now put IL-18 in the human system, caspase-4, 5, 11. We now put IL-18 as a downstream target of LPS-activated caspase-4, 5. And if the pathway is overactivated when during bacteremia or septic shock, or if you give the mice really high dose of LPS, peritoneal injection of high dose of LPS, the mice will undergo endotoxic shock or septic shock. And if you knock out caspase-11, the intracellular LPS receptor or the downstream pore-forming protein gastromin D, and you block the septic shock. So this clearly suggests that gastromin D is a promising drug target for treating sepsis. So during sepsis, the patients in the ICU, they can be recovered by other means. However, some of the patients will develop a disease called sepsis-associated encephalopathy. And this, many clinicians are aware of this disease. How the development of SAE or sepsis-associated encephalopathy is thought to be due to the disruption of blood-brain barrier structure during bacteremia or during bacterial infection or septic shock. So here is the structure of the blood-brain barrier. You have endothelial cells lining up with tight junction connected on the vessel in the brain. So we can model this BBB disruption during LPS-induced septic shock. You can see that we give the mice a little bit of a blue dye into the circulation. Normally, the mice have an intact BBB. The brain will not uptake the dye from the circulation. However, under septic shock, all the mouse brain turns to blue. So it's a very dramatic phenotype. And we also we can also show this using many other different BBB tracers that got the same results. But if you do that in the caspase-11 or gastromin D knockout mice, you completely block the LPS, high dose of LPS induced BBB breakdown. So it's the black and white phenotype or blue and white phenotype. It's very dramatic. So what is the underlying mechanism? So we first use electron microscopy to examine the structure of the blood-brain barrier. And you can see this is the large artery vessels in the brain, and the endothelial layer is already detaching from the basal membrane, and the dark nuclei are those cells undergoing pyroptosis. So the cells are shrink, the cells with condensed nuclei, and the cell body becomes shrunken, and of course the tight junction will be disrupted. And so we can clearly see the disrupted BBB structure in the LPS-shocked mice. And you can see all those endothelial cells, their nuclei become dark. And we also look at whether it is the brain endothelial cells that are sensing LPS to undergo pyroptosis, right? So we did lots of profiling, including single-cell RNA sequencing analysis. So the take-home message is that here is the vessel. You can see that the endothelial cells were lining up on the wall of the vessel, blood vessel. You can see though by immunofluorescence staining, we show that those endothelial cells express a high level of gastromin D and overlap very well with CD31, which is a marker for brain endothelial cells. And if we isolate the endothelial cells, primary endothelial cells from the mice, and we give the cells cytosolic LPS stimulation, and the cells will uptake cytotoxic green from the extracellular space and undergo pyroptosis. About 40% of the cells basically will die out of pyroptosis under in vitro conditions upon cytosolic LPS stimulation. And if you do that in caspase 11, knockout primary brain endothelial cells, you don't see that. By a Western blot, you can clearly see gastromin D cleavage by LPS activated caspase 11 into the pore-forming active form. And this was not seen in the caspase 11 knockout brain endothelial cells. So here, the phenotype I show you, the blue-white phenotype I showed you is the total knockout. So we don't really know, we don't have a definitive evidence that it is the brain endothelial cells that are sensing LPS in the circulation. So to address this question, we took advantage of AAV system, and this AAV-BI30 is specific for targeting brain endothelial cells, not other parts, not other endothelial cells in the body, only brain endothelial cells. So with this AAV system, we can use the AAV to express gastromin D and to express, to rescue the phenotype in the total knockout, right? So in the total gastromin D knockout, LPS-induced BBB disruption was blocked. However, if we re-express wild-type gastromin D specifically into brain endothelial cells, we can restore the BBB disruption. If you do that with the cleavage-resistant mutant of gastromin D, you cannot restore the BBB breakdown. And here are the statistical data. And we also take advantage of the AAV system to express a Cre enzyme, using the AAV to express the Cre enzyme in the floxed mice, so that we can also achieve, at the protein level, we can also achieve a specific knockout of gastromin D in the brain endothelial cells. We can also, you can see that the conditional knockout can block the BBB disruption. So then one of the reviewers really hate the LPS shock model, really wanted us to see the bacterial infection can trigger this pathway to induce gastromin D activation and BBB disruption. But we tried 10 different bacterial infection models, but none of them worked. And the paper was rejected, and it was in my computer for more than 1 year. And one day, and we realized that mice are much less sensitive to LPS than humans, right? This is well known. And indeed, Professor Joseph Buxbaum's group, back to 10 years ago, made a caspase-4 transgenic mice, basically humanized the cytosolic LPS receptor. And they showed that these mice are much more sensitive than wild-type mice to LPS. So we confirmed this in this lethality assay. You can see that in the wild-type normal mice, 50 milligram per kilogram LPS will kill mice within 1 day. However, for the caspase-4 humanized mice, you only need 5 milligram. So 10 times lower the lethality, 10 times lower LPS dose for the lethality. So using this humanized mice, and we can show that in this Klebsiella pneumonia infection model, which is mainly responsible for hospital-acquired infection, is also part of the sepsis inducing bacteria. So using the humanized mice, we can see that Klebsiella pneumonia infection can cause BBB disruption. Here are the statistical data. If you cross the transgenic mice with gastromin D knockout mice, you block the BBB disruption. Here is another assay using a more sensitive BBB tracer. We got the same results. So during the revision, we also found a paper coming out, a very interesting paper came out from Professor Florence Schmidt's group at the University of Bonn. And they made a nanobody for gastromin D. Not only for recognition, the nanobody can neutralize the pore-forming activity. Basically, at the protein level, using the nanobody, you can neutralize the pyroptosis or block pyroptosis by neutralizing the gastromin D pore-forming activity. So we came out with the idea we can use the AAV system, right? The AAV we have is specific for brain endothelial cells. We can use the AAV to express the nanobody so that the nanobody is only expressed in the brain endothelial cells. So we did that. We can see that if we express nanobody against the gastromin D, we can block LPS-induced BBB breakdown. And in the transgenic or caspase-4 humanized mice with Klebsiella pneumonia infection induced the BBB disruption, the nanobody expression in the brain endothelial cells can also block the BBB breakdown. So here is a summarized model of this part of the story, that when you have bacteremia or bacteria gets into the circulation, LPS is complex with LBP, which is very well known, and the LPS will be endocytosed into the cytosol through the CD14 pathway. And I did not have time to show you if you take LBP or CD14 knockout mice, we can also can block LPS-induced BBB disruption. So a intracellular LPS will then be recognized by caspase-411, and then the active caspase process gastromin D to liberate its pore-forming domain and making pores in the plasma membrane. So the pore is giant, 22 nanometer. Of course, you will get a material exchange across the endothelial layer. Under extreme conditions, those endothelial cells will die out of pyroptosis. So I've told you that this non-canonical pathway, caspase-4, 11 recognizes cytosolic LPS cleave gastromin D, and in the case to drive pyroptosis, in the case of human system, the active caspase-4 will also process interleukin-18. So this pathway operates in epicellular cells and also in brain endothelial cells. Of course, the pathway will also work in myeloid cells. And for gastromin D is a pore-forming protein. In human, there are 5 gastroamines, A, B, C, D, E. So each of us have 5 gastroamines. They share this 2-domain architecture and with auto-inhibited structure normally. So gastromin D is activated by the inflammasome, caspase-1, caspase-4, right? Canonical, non-canonical inflammasome. We have been actively working on 4 other gastroamines, and those gastroamines, they are not activated by inflammasomes. I'll show you one case. So those gastroamines, their activation is inflammasome independent. And quite a few years ago, we showed that gastromin E, another gastroamine present in human and mice, is activated by caspase-3. And caspase-3 cleave this linker region to liberate the N-terminal pore-forming domain. And the gastromin E is highly silenced in cancer cell lines. So 80% of the cancer cell lines, they don't express gastromin E. So if you activate caspase-3 by DNA damage, those cells will undergo apoptosis because caspase-3 was used to be used as a apoptosis marker. However, and those are primary cells, not cancer cells, like placenta epithelial cells, epidermal keratinocytes. If you induce DNA damage to activate caspase-3, those cells will undergo pyroptosis. So in collaboration with Professor Hua Lancheng's group in Harbin, and we actually look at the gastromin E, the role of gastromin E in virus infection. specifically in H7N9 avian virus. So many virus infections will induce caspase-3 activation. If you look at the literature, it's very common. So in this primary lung epithelial cells or some alveolar epithelial cell lines, we infect with H7N9. We can see caspase gastromin E cleavage, very robust, and caspase-3 cleavage. You can see that those infected cells by H7N9, they undergo pyroptosis with bubbles blowing out, right? And in the mice, we can also show that in the mice infection model, the mice lung has immune cell infiltration, bronchialization of the alveolar. So we can, if you look at, if you stain caspase-3, you get a caspase-3 positive signal. You can do Western blot, you get caspase-3 activation in response to H7N9 infection in the mice tissue. And the gastromin E is also gets cleaved, gets activated. So H7N9 infection in mice, like in human, is very severe, much more severe than COVID. And the lethality is almost 50%. 50%. Back to 10 years ago, there was a small endemic in Shanghai. And the mice, upon infection by H7N9, also die out of cytokine storm. And we show that using gastromin knockout mice, H7-induced N9-induced multiple cytokines were blocked. And his histological data also confirm this. You look at the body weight change, wild-type mice, the body weight decreased, and the mice died. And the knockout mice also have body weight decrease, but then they can recover, and they stay alive. It's a very dramatic phenotype. So the company I founded called Pyrotech developed a specific inhibitor for gastromin E, will be in clinics next year. And this small molecular inhibitor for gastromin E gave the same kind of results. You can see that if you treat the mice with the inhibitor, the blue line, the mice has body weight change but then recovered, and most of the mice stay alive, unlike the vehicle-treated mice. So suggesting that if we have another endemic with H7N9, and this inhibitor could be an effective drug to save people's life. And afterwards, there are several papers coming out showing that the gastromin E pathway is involved in viral infection. John Kagan's group at Harvard showed that vacciноvirus infection in the skin can induce gastromin E activation. Professor Jin Ye's group in China also showed Zika virus can cause placental pyroptosis and associated adverse fetal outcome by activating gastromin E. So gastromin is not only present in mammalian system, it's also present in unicellular organism. So like in bacteria, some bacteria also contain encoded gastomins, and they get cleaved by a protease and form very large pores. In bacteria, the pores formed to kill the bacteria is function to in antiphage defense. And in the fungal system, and this will work from others, I just want to show you. In fungus, there's a phenomenon called error recognition. And if two haploid streams are genetically incompatible and the cells fuse together, the fungal gastromin gets activated. The fused cells die out of pyroptosis. So this suggests that the pyroptosis is probably the most ancient form of cell death existing in unicellular organisms. In the case of bacteria, it also gets cleaved by protease. Fungi has two systems. One is proteolytic cleavage, and the other is very interesting. It's called RCD1 in Neurospora. In Neurospora, it only contains gastromin N domain, no inhibitory domain, suggesting they're not activated by proteolytic cleavage. So it turns out that the Neurospora has RCD1, a polymorphic RCD1. The genetically incompatible strains will encode RCD11 and RCD12. They're highly polymorphic, and the protein is about 95% of homology. And the two proteins will dimerize, will heterodimerize upon cell fusion, and the heterodimer will serve as a unit to oligomerize into a pore, RCD1 pore, and to kill the misfuse the fungal Neurospora strain. And in another organism called Placosa, and this organism also encodes a gastromin N domain protein without an inhibitory domain. And in this organism called Trichoplex adherens, when we found that this protein only contains a gasdermin and pore-forming domain. And this protein forms a dimer at the inactive state. The dimer is linked by disulfur bonds. Under redox conditions, the disulfur bonds get reduced, and then the monomer become active to oligomerize into a very giant pores, and 44 subunits and 28 nanometer. So this highlights that in those ancient eukaryotic organisms like Neurospora or Placosa, and there are very unique cleavage-independent activation of gasdermins, suggesting that in those organisms there could be diverse mechanisms of autoinhibition and activation in the gasdermin family, superfamily of proteins. And I will end by acknowledging the student, postdoc fellow in my lab who did all this work. Thank you.
Okay, many thanks for Professor Shao's wonderful talk. Any question? Since the time and just 1 or 2 question slots available.
So, Dr. Shao, I have a very short question. So you have showed the gasdermin E inhibitor is effective against the H7N9 induced viral disease. Have you ever tested to other viruses like SARS-CoV-2 or other acute infections?
We have not. So the COVID in the mouse model, it does not, unlike the H7N9, the H7N9 in the mice infection very much mimic the cytokine storm in the human, but the mouse model for COVID actually is not that good. So we haven't tested that because we don't have peace reference facility. We have to collaborate with others.
It could be broad spectrum. Well, I think so we can help you to test this.
Sure, yeah, we'll be happy to do that.
Wonderful talk. I'm wondering, by knocking out gasdermin, if you stop the brain from ballooning, do you stop the mice from dying? In other words, how much does blood-brain barrier breakdown contribute to
Very good question. If you, in the knockout, you block the BBB, you completely block the BBB disruption, but the mice still die. The mice still die, yeah. So the BBB disruption is not the cause, or is not the main cause for the lethality or high dose of LPS-induced lethality.
Okay, if no more questions, and many thanks again for
Thank
Professor Shao's talk. Okay, thank you.